Refinement is about minimising harm and promoting good welfare in animal research. This starts with husbandry, the way we house, handle, and care for the animals. Good husbandry, whether in the lab or in the field, improves data quality and reduces the risk of causing unnecessary distress. Refinement isn't just about ethical animal use, it's also about doing better science. Poor care or stress can distort insect behaviour, reproduction, and survival, both across and within generations. That affects the data collected and ultimately the robustness of our conclusions. Insect species are incredibly diverse, but there are some common areas where welfare can be considered, and practical steps reviewers can take to ensure scientists are addressing them. Refinement areas can include enclosure design, environmental parameters, nutrition and hydration, population density, health management, and colony maintenance. Enclosure design, where feasible, should consider the insect's natural behaviours, such as climbing, flying, burrowing, or hiding. For example, enclosures should avoid smooth, slippery walls for climbing species and provide species-appropriate substrates for burrowing species. Enough space and resources should be provided that each insect can perform its natural behaviours. Enclosures should be designed to reduce the risk of insect escape. Reviewers should always consider the species and research objectives. Work with invasive species, pests, and infected vectors may require special constraints. Insects are largely, although not exclusively, terrestrial and ectothermic. For terrestrial species and life stages, their survival depends on the temperature and humidity of their environment. For aquatic species or life stages, temperature, pH, and oxygen levels of the water may be important. In labs, these can be controlled. The proposal should demonstrate that these and other factors, like photoperiod, which helps govern circadian rhythm, have been considered and are within species normal ranges. When live trapping in the field, researchers should consider the ambient conditions in collection containers and adjust activities to avoid exposing insects to unintentional and inappropriate conditions that could impair their welfare. If detrimental conditions are likely, then monitoring may be necessary. Whether in the lab or in the field, researchers should ensure enough and suitable food and water is available. Practically, food or water may not be safe to provide during transit or in the field. If this is the case, collection times and species-typical feeding behaviour should be carefully considered in determining how long individuals will be left without food or water. If food and water aren't required at all, as sometimes the case for insects, researchers should explain why not. Plans should be sensitive to the risk of drowning and the challenge of reducing aggressive behaviour, which can occur in some species when food is provided in common areas. A clear plan for the monitoring and recording of such conditions can help standardise results. These can then be reported when publishing. Overcrowding can lead to stress, disease, aggression, and cannibalism. These can lead to poor research outcomes. In lab settings, researchers should explain how they will monitor for these issues and how to address them if they arise. Appropriate stocking densities are affected by many factors. For some species, such as highly eusocial species or larvae that aggregate to feed, low stocking densities can be as stressful as overcrowding. Also, there may be situations, for example in some large insectaries or commercial settings, where higher stocking densities are necessary for standardization. Unintentional disease and injury can impact the outcome of a study by altering insect behavior, physiology, and survival. In the lab, researchers should regularly monitor for signs of disease, including discoloration, unusual odors, presence of disease agents, lethargy, paralysis, and death. Injured or diseased animals may need to be humanely euthanized, as few methods are available for treating insect diseases at this time. Alongside proactive monitoring, cleaning insect enclosures with disinfectants and adding quarantine periods before introducing new animals can mitigate disease risk. Refinement doesn't end with insects actively enrolled in studies. Researchers will often rear more insects than they plan to use. This is to act as a buffer for experiments that don't go to plan, or for losses accounted during rearing. Just like with vertebrate models, insects bred or housed for research should be treated with consideration, even if they're not ultimately part of a published study. Having a plan for surplus or colony animals is thus part of responsible stewardship. Housing and husbandry standards should apply to all insects, not just the experimental ones. Cull surplus individuals humanely if they are not required. Plan ahead to balance rearing needs and avoid unnecessary overproduction. And assign responsible caretakers to ensure consistent colony maintenance, no matter the work schedule of an individual employee. In both a field and a lab context, refinement means being proactive. This means building in regular animal welfare checks into routines, SOPs, and colony maintenance schedules. In addition to good practice, such checks allow greater understanding of what causes stress in the research species, which can inform future studies and practices. Sharing such findings and reporting in a transparent way can help others in the field promote good practice outside of confidentiality requirements. Welfare check questions may include: are insects behaving as expected? Are feeding and hydration systems working, and is food clean and fresh? Are enclosures clean and safe? Are overcrowded or injured individuals being identified and removed? If you're reviewing an insect study, ask questions that promote thoughtful refinement in both lab and field settings. Key questions include: do enclosures address species-specific behavioural needs? How are temperature, humidity, and light managed? Are food and water required, and if so, are they delivered safely? How is population density being managed? Is the colony being adequately cared for? Is there a plan for monitoring insects regularly? Refinement is about respecting the lives and needs of all research animals. Whether insects are being used in a controlled lab or a field site, small adjustments in husbandry and handling can significantly improve welfare and data quality. In our next video, we will continue our focus on refinement, specifically looking at invasive procedures, anaesthetics, analgesics, and humane endpoints.